Vietnam National University of Agriculture, VNUA, and Utsunomiya University, UU, to this event, which is supported by the Sakura Science Foundation. And as people were just discussing before the meeting started, lots of people already have connections, and unfortunately, because of COVID-19, we cannot be traveling at this time to visit each other, but we hope this can provide a good foundation for research, collaboration, and exchange going forward. So without further ado, I'm going to stop. Oh, I guess I'm going to stop sharing my screen. One other thing I would like to encourage, though, there is a chat function. So we probably will not have time for everybody to ask questions, but I encourage you, please use the chat function. Feel free to share your comments, any questions you may have. We may have some time, and there's also a direct messaging feature too. So without further ado, I would like to turn it over first to Dr. Kenji Yamane for some welcoming words.
Okay, thank you, Benjamin. Hello, everybody. Konnichiwa.
Konnichiwa,
I'm Kenji Yamane, Dean of School of Agriculture, UU. Thank you for participating in this joint seminar of UNPAD and VNUA and UU. I greatly appreciate Dr. Nono in UNPAD, who proposed this seminar at first, and he's also an alumni of UU. So, and I'm very pleased to that Dr. Hui and VNUA members kindly joined this seminar. I also thanks to the speakers, Dr. Luciana Daja and Professor Natsuaki Tomohide and Professor Wada, head of our Sakura Science program. UNPAD and UU have MOU in the field of geology and agriculture since 2016. And VNUA and UU also have MOU, of course, in the field of agriculture since 2018. Now, as a global climate change becomes more apparent, it's very significant to be able to hold this seminar on themes related to SDGs. We are confident that we will be able to learn a wide range of topics from advanced science to social issues, such as plant viruses and forest ecosystems and African agricultural development and agricultural business and economy. I hope that the Indonesian, Vietnamese, and Japanese and the US citizens participants who are gathering today will foster friendships and share their awareness to the SDGs and commit to the SDGs together in the near future. Today we have a very great speakers and great participants. So thank you very much.
Thank you, Dr. Yamane. Okay, next I would like to invite from UNPAD, Dr. Nono, for some welcoming words.
Okay, thank you very much for arranging this meeting. Ladies and gentlemen, good day. Konnichiwa.
Konnichiwa.
Konnichiwa. Thank you very much for Professor Yamane and also Dr. Wada and maybe other sensei from Utsunomiya University and also from Vietnam Agricultural University. I'm sorry I could not mention the name. And also thank you also for our distinguished moderator, Dr. Benjamin. Thank you very much for arranging this meeting. And thank you very much also for distinguished speaker, Professor Natsuaki and Dr. Luciana Jaya. Thank you very much for what we call preparing for material for this joint seminar. We do hope we can strengthen our collaboration for the near future. And first of all, I would like to introduce myself. I'm Nono Carsono from Faculty of Agriculture, Universitas Pajajaran, but we usually call UNPAD here, located in Bandung, the capital city of West Java. Maybe Dr. Yamane and other sensei, and also from Vietnam, please welcome to our university. And on behalf of our dean, Faculty of Agriculture, Dr. Mehdi Rahmadi, I warm welcome you to this very important joint seminar. We do hope that it will strengthen our excellent collaboration, and this meeting also important for our togetherness, our relation, in order to make a close collaboration between Sonoma University, Vietnam Agricultural University, on also Pajajaran, Universitas Pajajaran. In this event also provide our young generation, actually a student from our agriculture faculty, our agriculture study program, to share their knowledge and perspective also for improving crop production to fulfill ever growing human needs and also SDGs. That's all our speech sensei and all. At the end, we would like to thank you for everyone who arranged this meeting, and thank you very much. That's all. Thank you, Dr. Benjamin.
Thank you so much for your kind words and for all of your help in organizing. This is a really exciting event. Okay, so next I would like to invite for some opening words from Vietnam National University, VNUA, Faculty of Agriculture from the Department of Plant Pathology, Dr. Nguyen Duc Huy. And sorry if I mispronounced your name, but please.
Thank you very much. Thank you very much. Good afternoon, ladies and gentlemen. First of all, I would like to give my deep thanks to Professor Natsuaki, Dr. Yamane, Dr. Wada, Dr. Nishira from Utsunomiya University to invite me and also our students to join online seminar for Sakura program. Also, on behalf of our faculty and university, we would like to thank all of you so much. Even this time, we have big problem for COVID-19 spread everywhere. Also in Vietnam, we have big problem for 2 months ago. And all students and lecturer have to do most class online, and we do good online class this time. And even today, I asked more than 40 students join with us. So in 2019, we are very happy with your help, we can came to Utsunomiya University twice for Sakura Science Exchange Program, and our students can learn a lot from you, including culture, agriculture, and the other. Up to now, we always and still keep contact with you, not only for exchange student, but also for research in my site. So we expect that the COVID-19 will be recover soon, and all the people from the world back together, and we can connect all our collaboration for not only exchange program, but also research in agriculture field and the other field we have in our university. And from my deepest thanks to all of you, and hope near future we have chance to see you again. And in my case, I sometimes contact with Professor Nachiaki Sensei and also Dr. Nishira for plant virus research. And today I'm very happy to see two topics related to plant protection, especially plant disease and plant virus. And I hope in my for my side and also my student, because today all my student came from English class, third 2 year, second year, and third year student, and I hope even they take my lecture in plant disease currently, so maybe they can understand the lecture from you, and this very useful for us. So we hope that we can continue for our research in the field of plant disease and agriculture field also. And thank you very much again.
Yeah, Dr. Nguyen, thank you so much for sharing your words. And yes, the challenging time during COVID. And you mentioned your students are joining. I was impressed to see we have 464 participants right now. So many people are joining, so really exciting. Okay, next I would like to invite Professor Wada to share some words about the Sakura Science Program.
Thank you, Chairman. Good afternoon or good evening, everyone. I'm very glad to join this international seminar held by Universitas Pajajaran, Vietnam National University of Agriculture, and our Utsunomiya University. I wish to thank Dr. Nono and Dr. Jaya of Universitas Pajajaran, Dr. Phui of Vietnam National University of Agriculture, and Dr. Yamane, Dr. Natsaki, and other staffs of Utsunomiya University for their effort to promote this joint seminar. Last year, in 2020, we applied Sakura Science Plan to invite Indonesian and Vietnamese students of these universities to Japan and successfully obtained the fund of JST, Japan Science and Technology Agency. Sakura Science Plan is a short-term invitation program to give the aspiring youth opportunities to experience cutting-edge science and technology. However, as you know well, because of the COVID-19 pandemic, we could not invite students from Universitas Pajajaran and the Vietnam National University of Agriculture. As an alternative remote online project, today, this international joint seminar is taking place. This is very important to strengthen friendly relations among 3 universities. However, seeing is believing. I hope after COVID-19 season, we want to invite students from Universitas Pajajaran and Vietnam National University of Agriculture by Sakura Science Plan again. Thank you for attention.
Thank you,
Thank you.
Professor Wada. Okay, so moving on, we are coming to our first lecture. So I would like to introduce Dr. Luciana Jaya. She's from Universitas Padjadjaran, UNPAD, where she is a faculty of agriculture in the Department of Plant Pests and Diseases, and also at Functional Nanopowder University Center of Excellence. She researches cutting-edge technology about plant protection and the use of technologies such as silica nanoparticles. Her talk today is Biocontrol Delivery Systems using nanocarbon as a carrier in plant pest and disease management. So without further ado, I'd like to welcome Dr. Luciana Jaya. If you're able to share your screen, please do so.
Thank you, Dr. Benjamin Schreger. I think, yeah, I would like to share my own screen. So, it is.
I will let you know. Right now, I cannot see it. I think you
It's
are
loading.
designated as a co-host. Okay, I can see it now.
Okay, thank you. Shall we start now?
Yes, please.
Okay. Good afternoon for Indonesia and Vietnam time. This afternoon, I would like to share some studies in our laboratories. But before that, I would like to thank all the, let's say, committee or whoever who arranged this joint seminar. It's nice to have all of you from Universitas Padjadjaran, Utsunomiya University, and Vietnam. Oh, the one from Vietnam, Vietnam National University of Agriculture. Okay. Our studies about new or novel biocontrol delivery system using nanocarbon as the carrier in the system. I am from Department of Plant Pests and Diseases, Universitas Padjadjaran, and also a team member of the unit in Universitas Padjadjaran, which is called Functional Nanopowder University Center of Excellence. These studies actually part of the ALG or Academic Leadership Grant of Universitas Padjadjaran, and our group is led by Professor Dr. Hersanti, and the team member are Dr. Nur Istifada, Dr. Mika Setiawati, Professor Imade Joni, myself, Luciana Jaya, Dr. Sri Hartati, Rika Meliansyah, and Dr. Lindung Tripuspasari. We are all from various laboratories in Universitas Padjadjaran, and I myself and Professor Hersanti are the team member in Functional Nanopowder University Center of Excellence. The background of our study is about how to have environmentally friendly pest management. As we know, in plant cultivation, we change the natural ecosystem into agro-ecosystem. So we have to be careful with the ecosystem. We want to have a good or a sustained agriculture environment. But of course, in every plant cultivation, the constraints will be pest and disease, plant pest and disease. And then that's why we have to manage something to eliminate or to reduce the this pest and disease, so we can have a good crop quantitatively and qualitatively. In plant pest management, we have to choose the best method, of course, and one of the best methods is biological control, because it is more friendly to the environment, safer, and maybe easier to apply. And as the biological agent, we all know that we use predators or parasites or parasitoid. In plant pest and disease management, mostly we use microbes to control the pest. And these microbes we choose, there are some antagonistic microbes to pathogens, what we call antagonists. And also we have entomopathogenic microbes. And those microbes can be formulated in the form of biopesticide. In a biopesticide, we can have one microbe or maybe more, several species of the microbes, what we call consortium. So in our study, we have several different microbes that we can use as the biocontrol agent. And in the biocontrol delivery system, we use this nanotechnology, the technology that use nano-sized particle to produce the formulation. Why we choose this? Because we want to have a more simple handling of the pesticide, higher efficacy, and also we want to have a longer shelf life of the biopesticide. Now, mostly the farmer use the microbes, the antagonistic microbes, even the entomopathogen. They just cultivate or culture the microbes and then apply it in the field. But of course, the microbes cannot live for a longer time, only for a few days or even a few weeks, okay. But now we want to have this formulation, and we want to keep it longer. And then in a formulation of biopesticide, of course, the main ingredient or the sorry, I lost the word, but it doesn't matter. Of course, the microbe as the main ingredients, as the active, oh yeah, I got it, as the active ingredients. And then we should have also a carrier. And it is good if we can also add something else that can improve the quality of the formulation. In our study, we use carbon fiber as carrier, but smaller particles are always better because it is stable and has larger surface. So we chose the nano-sized carbon fiber. And in our study, we use graphite as the carbon fiber. In the carbon fiber was in graphite form. As we know, carbon has a lot of different form in even in nature or in the synthetic one. And also in our study, we add some plant nutrients in the formulation. In the beginning, we had silica in nano size, also zinc and titanium. But by the process, then we only focus in silica nanoparticles. Actually, in the formulation, we can also add some plant growth promoting agent. But of course, they are all should be matched, and all the microbes do not affect each other in the formulation. This is why we use nanocarbon. This is the figure about how the carrier carry the main ingredient or the active ingredient in a formulation. And I think what we have in our studies is this one. Oops, sorry, wrong way. And this is a diagram of the look or the simple diagram of the mixture in the formulation that we have. First, we have this carbon nanofiber, the suspension, and also we have the suspension of the antagonistic microbes to plant pathogen, or maybe the entomopathogenic fungi, and then we mix them. And the bacteria here will be encapsulated by the fiber, the carbon nanofiber. If we want to add something else, let's say silica nanoparticles, we can mix them. So later, in the end, the look will be like this in the picture. So we have nanocarbon fiber, and then we have the microbes here, and also we have this silica nanoparticle or something else, maybe a zinc or titanium oxide nanoparticle. And this is the apparatus to synthesize the carbon nanofiber. We have this equipment in Finder Co. This is the name of our unit, study unit in Universitas Padjadjaran. And this is the result of scanning electron microscopy of the carbon nanofiber. The two first picture here is 5,000 times enlargement. This is 10,000 times, and on the bottom, they are 20,000 times. Looks like that. And this is when we have the formulation. We have several different formulation. The first one, we have Pseudomonas fluorescens as the antagonistic agent to some plant pathogenic fungi and bacteria. Of course, we put the carbon in the form of graphite, and we also add the silica nanoparticle. And this is 5,000 times. In the middle, we have this different antagonistic bacteria. This is Licinibacillus. And on the right picture, that is the Bacillus subtilis. This is also antagonistic bacteria. So this is the resume or the what we are doing. What we have done, and we are still doing it now in our study to develop a biocontrol delivery system by using carbon nanofiber. The biocontrol agent, we have several different agents here. Antagonistic bacteria are Pseudomonas fluorescens and Bacillus subtilis. We have two different isolates of Bacillus subtilis. One is plant growth promoting rhizobacteria, and the other one is the endophytic bacteria and Lisinobacillus. And also we have here antagonistic fungi, Trichoderma harsianum, and entomopathogenic fungi, Metarhizium anisopliae and Beveria bassiana. As the active ingredients, we have these microbes, single or consortium of biocontrol agent. Why we use consortium? Because they have different mechanism. Like here, we can use the plant growth promoting rhizobacteria, which is the mechanism is to improve or to induce the growth of the plant, so it can be stronger against the pathogen. And also we can use Bacillus subtilis, which is endophytic, because they live endophytically inside the plant organ. And then maybe we can also use Trichoderma mixed with Pseudomonas fluorescens, let's say, because Trichoderma is a soil inhabitants, so they can prevent the infection of soil-borne plant pathogen. While Pseudomonas fluorescens also, actually Pseudomonas fluorescens also soil inhabited, but if we use endophytic Bacillus subtilis, so this endophytic bacteria can prevent the infection from inside the host plants. And then the formulation, also we use carbon as the carrier and edit with silica nanoparticles. Several plant pathogens we have studied, including Ralstonia solanacearum, the cause of bacterial wilt on several different plants, Sclerotium rosei on soybean, Fusarium oxysporum also on many different plants, including tomato, potato, and shallot. Phytophthora infestans, especially on potato, it causes late blight, and Alternaria pori and Pellicularia oryzae. Pellicularia oryzae is on rice. And also we have two pests. We have studied two pests, that is Spodoptera litura and Mysus persicae on potato. The crops, for the time being, we have studied potato, tomato, shallot, spring onion, and rice and soybean. The application method, this is actually crucial because how we apply the formulation, it also causes different result on the different plants. We use seed coating, of course, soybean, rice, and tomato, they are true seed. But for potato, we use the tuber seed. And then also we just shower the soil around the plants. And then also we spray the formulation on leaves. Now the results. From the results, we can make a concise result here that the biocontrol agent can survive in the formulation. Of course, they are viable in the formulation, and some can survive up to 18 months. How can we find that survival? Well, we just culture the bacteria or the fungi after we keep them in the storage for some period of time. And then we count the population. We measure the population. And the application method also, we study about this. All the plants by this method, seed coating, it seems have different response. For potato, the potato tuber, when we submerge the potato tuber in the formulation, it is okay. Then the germination or this it's good. But for tomato and rice, when we do this seed coating, then the germination reduce. So we think that maybe the carbon has some effect on the germination, on the seed germination of rice and tomatoes, because the seed of rice and tomatoes are very, very small. Then in the application, we cannot only apply the formulation once by seed coating, but we should repeat the application by soil drenching and leaf spraying to have a better result. The antagonism between the antagonistic bacteria and the pathogen, we do it both in vitro and in planta. In the in vitro experiments, we measure the colony diameter, and also we measure the inhibition zone caused by the antagonist. Of course, this is on the colony of the pathogen. And in the in planta experiment, we measure the incidence, the disease incidence and disease intensity, and also the incubation period. Most of the formulation cause longer incubation period. That means the biocontrol agent can delay the infection and also reduce the disease incidence and disease intensity. And about the entomopathogen, the formulation cause the mortality of the pest, increase the mortality of the pest. That means our formulation work well. I have some random, or I chose the data randomly to share in this seminar. This is about the viability test. We measure the population in the suspension, in the formulation, the population of the biocontrol agent. And after some time, they are still viable. That means all the microbes can still grow when we subculture it on the proper media, growth media. And also we have this survival test. We measure the bacterial population after quite some time. The longest one is 18 months. And some formulation, the biocontrol agent still live well. That means when we subculture it, it grows well on the medium and the population is quite high. And in the in vitro test, you can see here the inhibition zone. So on the background is the colony growth of Ralstonia solanacearum, the bacterial wheel pathogen. And this is the filter paper disc, which we have put the filter paper disc in the formulation for quite some time, about 30 minutes, and then we put it on the medium, on the agar medium. And actually the formulation caused this inhibition zone. That means the biocontrol agent can reduce the growth of the Ralstonia solanacearum. This is also the in vitro test by using the trichoderma against plant pathogen. The pathogen here is the trichoderma colony, the darker color one, and the white colony is the Fusarium oxysporum. And the growth of the Fusarium oxysporum is retarded. Actually, we have also some picture how the trichoderma growth on the Fusarium colony, and the mycelium like twisted the hyphae of the Fusarium. And in the plants, this is the example of late blight of potato caused by Phytophthora infestans. And the disease intensity was reduced by application of the formulation with carbon fiber. This picture shows how the formulation attach or adhere to the surface of the seeds. This picture is on the potato seed, potato tuber. On the potato tuber, we can see the flat angle here that show that the formulation can spread well on the surface of the potato tuber. That means the colonization of the biocontrol agent will be good on that kind of surface. And we have also measured some plant growth parameter like plant height, number of pillars for rice, and also number of leaves for potato and soybean and shallot, also the length of root. And we can see that all the formulation do not reduce the growth parameter. That means that the formulation we have with carbon fiber do not affect the growth of plants or do not retard the growth of the plant. So the conclusion is, the conclusion is that the biocontrol delivery system with nanocarbon fiber is promising to develop. We can see from these reasons, because the viability of the bioagent is well maintained and the biocontrol agents survive well in the formulation, and the mechanism, the in vitro mechanism of the biocontrol also increased significantly. The nanocarbon did not affect the in vitro growth of the biocontrol agent. I didn't show you the picture, but all the biocontrol agent grow well in the media added with nanocarbon fiber. And also the nanocarbon fiber did not have bad effect on the in planta mechanism of the biocontrol agent. So all the biocontrol agent can perform well, even if they already in the form of the formulation with nanocarbon. And also the formulation can well adhere to the plant surface. And we have never seen the toxic effect of the formulation on the plants. There are no toxic symptoms on the plants applied with this biocontrol delivery system with nanocarbon. And that's why we can say that nanocarbon is a good carrier in a biopesticide formulation. I think that is the end of my presentation. I wish to have some feedback from the audience, especially those who deal with plant pest and disease management. Thank you very much. I give the time back to the moderator.
Okay, thank you very much. Let's see, I had you on Spotlight. I'm not sure if you are still on Spotlight for other audience members, but I can figure that out. Okay, shall we start with the question from Dr. Yamane? Would you like for me to read it, or you could also say it with your own words?
Please read, Dr. Benjamin.
Okay.
I'm trying to read it. Okay, but you can read it for all of us.
So, yeah, the question, which is one I was thinking about too, it's such a fascinating talk about this cutting edge technology. Dr. Yamane writes, it has many applications and is a wonderful method. Can it be put to practical use in terms of cost so far?
Okay, thank you for the question, Yamane Sensei. We plan to do some study about the cost, but so far, as the carbon fiber is produced, let's say manually in the lab of Finder Yukoi, so the cost is quite high. But once we establish with all the work, maybe we can have a cooperation with some company to produce this carbon fiber in a lower budget, perhaps. We hope that, and also the application is quite simple, similar to the chemical synthetic pesticide. Just spray it, and even the storage, maybe we can put in a bottle, because we choose the liquid form of the formulation. We can just put in a bottle, and then any kind of bottle. It doesn't have to be glass bottle, because as we know, some synthetic chemicals can react with plastic bottles. So we can use any kind of bottle. And I think it is applicable in the farmer situation or condition in the field. Thank you for the question.
Okay, thank you very much.
I hope it can answer your question.
Yeah, that was a great answer. We don't have other questions from the chat. I was wondering, maybe Dr. Nguyen Ducoy, you said this is very close to your field, so you might have a question or a comment you could share.
Okay, thank you very much. First of all, I would like to thank a lot for your presentation on the biocontrol of plant disease. I work a lot of, I use a lot of trichoderma and also Chaetomium and also Bacillus for biocontrol in Vietnam. In my lab, I use at least 5 species of trichoderma and also 3 species of Bacillus and 5 species of Chaetomium. And as I know, this own way apply for controlling soil-borne disease like Fusarium, Rhizoctonia, and the other soil-borne pathogen. However, I would like to know that in your university or in your country, do you have any commercial product of trichoderma or the other ejection liquid? Some chemical ejection from trichoderma or from Bacillus apply as liquid for spraying to the crop. Yes, that's all. Thank you very much.
Yes, thank you for the question, Dr. Nguyen Ducoy. Yes, in Indonesia we have this product commercially the Bacillus and Trichoderma. But I think most of the farmer still use the, let's say, homemade biocontrol agent. So even in our lab, we can prepare some culture of this Trichoderma and Bacillus and other biocontrol agent because we have the isolates. And if the farmer needs, they can just order and then we cultivate it, we culture it for several days, and then we mass culture it and then give the culture to the farmer. That is the way now. But of course, the commercially products is also available. I'm not sure, perhaps someone here in our room, perhaps Professor Tarkus and Professor Helsanti can help me with this question. If the biocontrol agent with this Trichoderma and Bacillus is commercially available. I know that Bacillus thuringiensis, the pest bioagent, is available commercially, but I'm not sure about Trichoderma and other biocontrol products, biocontrol agent.
Yeah. So I think it's very important for us to use this antibiotics fungus to control soil-borne disease, because I'm also working on banana disease of banana.
That's right. Yeah.
Yeah. Because in 2019, I saw one publication from your country of banana disease, and see so very good classification of Fusarium now. I mean Fusarium oxysporum infected banana now. It's very good publication. I know in 2019 in your country, and it changed a lot for the classification of Fusarium oxysporum as banana disease of banana in your country. Yeah.
Yes.
So thank you, and maybe I to save the time, I maybe contact with you later for more discussion. Yes, thank you very much.
Yes, thank you, Dr. Nguyen.
Okay, so yeah, that was really fascinating discussion. I think in the interest of time, we'll move on to the next talk, but I'd like to again express my thanks for such a fascinating talk.
Sorry, I have question, Dr. Benjamin.
Okay.
Can you read in the chat? In the chat.
Excuse me, Dr. Benjamin. There is also in the chat a question from Kaina from University of Utsunomiya.
I see. Okay. Well, let's go through these questions and then proceed. But if it goes on too long though, we may just move ahead. But okay. So Dr. Nono, do you want me to read your question, or would you like to say
Yes, Dr. Benjamin, please.
Okay. So Dr. Nono writes, in pest disease application, some issues are found. For example, how to make this particle is more environment and not harmful to human. How to reduce toxicity, improving long life.
Okay. Thank you for the question, Dr. Nono. As I said before, that the formulation with carbon nanofiber is environmentally friendly because carbon is an inert material. Carbon doesn't react or with other material in the environment, and also carbon is quite stable. So it will not be harmful to plants. Oh yeah, sorry. You mean to human? Okay, to plants it is not harmful because we have proof that the plants grow well even when we add or when we apply this carbon nanofiber. But to human, I think it is also maybe, in my opinion, it will be safe because this carbon, graphite, or even in other form of carbon, usually used as the toxic material absorbent. What do you call it? Like when you have this stomachache, so to absorb all the toxic material in your stomach, we can use norit or carbon active to reduce the symptom of the stomach ache. So I think carbon is not harmful to human also. And then how we can reduce the toxicity? Perhaps we can use just the very the small amount of carbon or the very low carbon concentration in the formulation, as long as it is still it still work as the carrier. And also there is two more questions from Kaina in
Yes,
Yeah.
yes.
University of
Kaina-san. Kaina-san from Malaysian student.
Oh,
so-
from Malaysia.
Yeah, so other country student.
But this is you.
Yes.
Shall I answer this, or you would like to
Sure.
read the question first, Dr.
Yeah.
Benjamin?
Please go ahead. Please answer.
Okay. The method, seed coating, soil drenching, and leaf spraying. I think we have to combine all the three methods in the field, because before the plant grow, we have to prepare the seed. So the biocontrol agent can colonize the seed surface, and once the seed germinate, the biocontrol agent has already established in the seed. And then after we grow, if we plant the seed in the field, of course, there are a lot of obstacles from the environment. That's why maybe the population of the biocontrol agent can reduce. Then we have to apply some more by soil drenching. That is to prepare the new growth of roots to be colonized by the biocontrol agent. And the leaf spraying is actually for the airborne pathogen. And also maybe the mechanism, not directly as the, not directly to control the population of the pathogen, but more to increase or to induce the resistance of the plant against the pathogen infection. And the second question, yes, the survival of the biocontrol agent are variable in the formulation. The survival when we cultivate the biocontrol agent in culture media, because we prepare the complete or the nutrient for the biocontrol agent is quite plenty in the culture media. Of course, they will grow better than in the soil. In the soil, they have many there are many other things that can affect the growth of the biocontrol agent, such as the chemical condition of the soil, the pH, and also the other microbe which live in the environment, in the soil. Is that answer the question? Or maybe you have something else in mind about this question number 2?
think that answers the question. It's really great.
Okay.
Thank you very much.
Yeah, thank you. Oh, okay, again, everyone, please join with me in thanking Dr. Jaya for a really fascinating talk. And wow, we have over 500 participants. I did just want to acknowledge someone from UNPAD put in Lillian Rizki put in a link for attendance. So I think maybe for UNPAD students, they want to be sure to click this link. Okay, so now I want to start to transition towards our second lecture of the evening. And I would like to introduce Professor Emeritus Tomohide Natsuaki, who is based at Utsunomiya University as a specially appointed professor and senior coordinator for Inter-University Exchange Project, Promotion Center for International Exchange. And his talk today is fighting against plant viruses to save food production. And without further ado, I will turn it over. I see you're already sharing your screen. Great.
Okay. So the PowerPoint is okay? No problem.
Yeah, it's looking good.
All right.
Yeah.
And my voice
Okay,
is
sensei.
also okay. Sound is enough.
Yeah, the sound is okay. There's a little bit of humming background noise, but it's okay.
Yes. Anyway, so thank you very much for your invitation to this seminar. So first of all, I would like to express my deep gratitude to all those who are contributing to this seminar. Dr. Nono and colleagues in Pachachala University, and also Dr. Hoi from Vietnam National Agriculture University, and staffs in Utsunomiya University. So anyway, so by the COVID-19, the world is changed drastically. And for example, 2 years ago, I have visited Indonesia, Pachachala University, and talk about the same things. However, at that time, so I did not image the web lecture. However, the COVID-19 changed the world, and we can do the seminar or lectures on web with internet. So the distance is not problem in education. So anyway, yeah, because we have the international exchange agreement with Vietnam National University of Agriculture in 2018 and Pachachala University in 2019. And I have visited both universities. So I think I'm the only person who visited both university directly, but now we cannot. Anyway, so the number of audience is more than 500, so I don't know actually the scientific background. So I exchange my PowerPoint. The first one is I would like to define what is virus. Yeah, because COVID-19 and also the plant viruses are just viruses. So the characters of COVID-19 and plant viruses are very, very common. So the first, we must understand that the viruses has no cells. Viruses has particles. Okay, and usually the cells of any organisms, including human beings, contains the nucleic acid of DNA and RNA. However, the virus contains DNA or RNA, not both. And also, the viruses cannot translate proteins or replication by themselves. So the virus must enter the cells of suitable hosts and hijack the gene expression systems. So the cells, invaded cells should be living. And also the viruses are not always pathogens. Some viruses cause no symptoms. And also the viruses is not so small. Some viruses are more than the diameter of some viruses are more than 1 micrometer, and we call them the Pandora virus. Anyway, so this is a summary of gene expression system in your body, not only in your body, but also in plant, animals, and so on, or fungi, bacteria. So originally, the DNA code the gene and transcripts to RNA, and from RNA translated to protein. So the coronavirus has one single strand RNA as genome. So the coronavirus cannot multiply by itself. So the coronavirus enter the cells of human beings and viral RNA. So multiply or amplify viral RNA using this gene expression system in human being cells. So the human being cells are misunderstood that the coronavirus RNA is human beings RNA and translate protein, so make a lot of protein, viral protein, and amplify the viral RNA. So after that, the cells release a lot of virus particles like that. So it's very difficult to control the viral multiplication in host cells directly. So I would like to talk about the cross protection because nearly 100 years ago, the cross protection phenomenon are reported with the tobacco mosaic virus. And in cross protection, the first virus infect plants, and that plant inhibit the multiplication of subsequent infection of another isolate of the same virus or closely related virus, the second virus. That means this phenomenon is very similar to the vaccination. And so many researchers reported about the cross protection in plant viruses like tobacco mosaic virus or potato virus X and potato leaf roll virus. So these viruses are RNA virus. And also the cauliflower mosaic virus. So DNA virus also shows the cross protection and single strand DNA virus. So some virus contains a single strand DNA as genetic molecules. So the single strand DNA virus also shows the cross protection. And also the viroid. Viroid is a naked RNA pathogen. So only very small RNA molecule are also pathogens. So there are many kinds of viruses shows the cross protection phenomenon. And so many researchers consider about how to use the cross protection for the control of virus diseases. So nearly 70 years ago, the first trial to use the citrus tricida virus mild isolate to protect the disease of orange trees. And also the mild isolate of cacao solen shoot virus was used to protect cacao trees. And still now, the citrus tricida virus as a mild isolate of citrus tricida virus is used to protect citrus trees. So the problem is immune and the vaccination. What is different? Yeah, because in animal cells, so usually the immune or vaccination means the production of antibody against the pathogens. However, nearly 100 years ago, so some researchers used the term immunity for plants, or also the vaccination. The term of vaccination is used for plants. However, the late of 20th centuries, the immunity and vaccination are not used so many times in plant science. However, in 21st centuries, the plant immunity are recovered, and many scientists reuse the plant immunity or vaccination. So we use the term vaccine or vaccination for plant viruses. And so what is immunity? So actually the narrow definition is make the resistance. And broad definition is, make the antigen. However, the broad definition of immunity is increase the resistance. So for example, the first speaker, Dr. Luciana, talked about the biopesticide. So that means many microorganisms can introduce the resistance on plants. So the attenuated isolate and cross protection are used to control plant virus diseases. So attenuated isolate is a protective virus and cause no symptoms or very mild symptoms on the crop. And the good attenuated isolate should show the high degree of cross protection efficiency. And so the cross protection is very similar to the vaccination. In Japan, so there are several examples to use a mild isolate of these viruses to control the virus diseases. So tomato mosaic virus or cucumber mosaic virus, pepper mild mottle virus, and so on, so on. So in our research, the first, I would like to talk about the zucchini yellow mosaic virus. So the zucchini yellow mosaic virus is a causal agent of mosaic diseases on cucurbit plants. And so usually the infected cucumber plants shows a mosaic, or fruits of cucumber shows a malformation. And finally, the infected plants shows a very wilt and died, like that. So cucumber mosaic virus is a member of potivirus. The virus particle is like this one. So the virus particle is a long filamentous particles. And the virus is transmitted by aphid. So it's very similar to the human viruses, because some human viruses are transmitted by mite or mosquito and so on. So the zucchini yellow mosaic virus is transmitted by aphid. So usually, so pesticides to control aphid are used to protect cucurbit plants from the virus infection. Anyway, so the zucchini yellow mosaic virus contains a single-strand RNA with 10,000 nucleotide sequence. So how to produce the attenuated isolate? So we have established the new technology, new selection procedure. So the first, the inoculated plants or infected plants with zucchini mosaic, yellow mosaic virus are stored in low temperature, about 15 or 12 centigrade. And then the mosaic symptoms shows the green area and yellow area. So yellowing area contains a lot of severe virus particles. However, the green area doesn't contain any severe isolate virus. Green area contains only attenuated or mild isolates. So we cut green area and inoculate to new plants. So after sub-inoculations, the green area is enlarged and enlarged one by one. So finally, the old leaf shows no yellowing area. At that time, so we can establish the mild isolate like that. However, this procedure can be used only for sub-transmissible viruses. That is the weak point of this method. Anyway, so we have established a good attenuated isolate, and we inoculated this mild isolate to cucumber first, and then inoculate with severe isolate. So this one, the center one, the first inoculated with attenuated isolate, and then inoculate with severe isolate. So the center one shows no symptoms, very similar to healthy one. However, without first attenuated inoculation, so the plant shows the severe mosaic symptoms of severe isolate. So finally, we can get a good attenuated isolate like that. So the most important is the field trials. Sorry. So the next stage is we have produced the ampoules like that. So in the COVID-19, you can watch the ampoules of vaccine solution. So this is very similar one. However, this is the dried one. So when we first we resolve the dry one, and then inoculate the leaf. So this is the manufacturer procedure to produce the freeze-dried ampoules like that. Anyway, so this is the commercial company's job. So we can buy the ampoules contains the freeze-dried extract of attenuated isolate, and resolves with water and sub-inoculate to seedlings like that. So the inoculated cucumber seedlings multiply the attenuated isolate and show the resistance against the severe isolate infection. The second problem is that mass production of inoculated seedlings, because we don't have good machines for sap inoculation, so that we must employ the many people for the inoculation of seedlings. So the price of one seedling is $1 more expensive than ordinary one. However, if there is no damage, so the non-inoculated plants, farmers cannot get any yield. However, the inoculated and vaccinated plants, farmers can get a lot of cucumber fruits like that, for example. So the farmers can get the benefit. So they can pay $1 for one seedlings. So the most, and this is a good example for the farmers. So the left side, all seedlings are inoculated with attenuated isolate, and the right side, there is no inoculation. So the farmers recognize that, oh, the vaccination is very, very useful, and they can get the good fruits, so it's okay. So this field trial is very important. And next is the molecular analysis of attenuated isolate. Because for example, you can, so in newspaper or TV news says that the lambda variants of COVID-19 has a specific mutation on T76I and so on. So completely same, but this mutation in COVID-19 change to more severe characters. However, in attenuated isolate, for example, there are 4 typical mutations on the amino acid sequence in H protein. So here, so the A350 changed to T, or L-leucine 636 changed to serine. So these mutations occurred on the genome of attenuated isolates. So these mutations cause attenuation in zucchini mosaic virus. And so there are 4 mutations which related to attenuation. So this is very important because in vaccination, so 50 years ago in Netherlands, they sell the attenuated isolate of tomato mosaic virus commercially. However, that isolate contains only 1 mutation. So soon the revertant, so the mild to isolate change mutated to severe one, and the farmers very surprised. Many tomato plants show the mosaic symptoms. However, in this case, zucchini mosaic virus attenuated isolate contains the 4 mutations related to attenuation. So this is very important because 4 nucleotide, 4 amino acids reverse to the original amino acid. So it's impossible to cause naturally. So we get the patent, and now the Utsunomiya University gets the benefit by this patent. Not so big. Anyway, so now this ampoules is sold by the Kyoto Biken company. So we are still working with them. And also this is another type of attenuation. So the cucumber mosaic virus. Cucumber mosaic virus is have the spherical virus particles like this, and the mild isolate cause no symptoms and protect tomatoes from the infection of severe isolate. So the left side shows the yellow and mosaic caused by the cucumber mosaic virus. However, the right side, there is no symptoms. And now the inoculated tomato seedlings are sold in spring season at the garden stores. And I'm sorry, so these are written in Japanese, but CMV vaccine inoculated seedlings. So maybe the Chinese or so Dr. Nono can also read this, I hope. Sorry. Anyway, so the next step for near future, the most important is the combination vaccination. So for example, the in fish, so there are many vaccines against the fish diseases. So these vaccines are against the bacteria and virus diseases. So the number of the vaccine is increasing drastically. So sorry. And now, oh my God, sorry. And so the problem is that the combination vaccine we must develop because many crops or many vegetables or fruits are infected with several viruses. So we must establish the combination vaccines against the plant viruses. So, and also the modern biotechnology developing the new approach like recombinant RNA vaccines, not only human beings, but also the fishes. So recombinant RNA vaccines, so Pfizer's vaccine against COVID-19, the recombinant RNA vaccine, like that. So maybe in near futures, the recombinant RNA vaccines are established for fish diseases, I think. So for the near future, we have many problems resolved for the vaccination of plants. The first is the inoculation method. So the sap inoculation is very, very laborious. So that we must a more simple and more efficient inoculation method. So we are considering about the vacuum inoculation. So the very small seeds or just the germinated seeds are soaked virus solution and vacuumed. So the plants absorbs the viral solutions and the virus can multiply in the plant. And the second theme is how to produce the combination vaccines. So, for example, the cucurbit plants, zucchini, not only zucchini yellow mosaic virus and the cucumber mosaic virus, but also the watermelon mosaic virus, papaya ring spot virus, cucumber green mottle mosaic virus, and so on. So now we have the trial field to do or to establish the combination vaccine. So maybe the Dr. Nishigawa, he will do that in the future, I hope. So, and the third one, so vaccines against non-subtransmissible viruses. So this one is a pepper yellow leaf curl virus in Indonesia. So the peppers, hot chili plants in Indonesia are infected single-strand DNA virus, like tomato yellow leaf curl virus or pepper yellow leaf curl virus, and so on. So these viruses are only transmitted by whitefly insects, and we cannot inoculate, if we can establish the mild isolate against these viruses, however, we cannot inoculate directly to plants. So that we must establish the new inoculation method as a vacuuming or something like that. And the next one is a genetically modified technique. So if consumers accept the transgenic plants as food, so the transgenic plant is more easy way to protect virus infection in plants. And so the integrated pest management, IPM, is a very important thinking route. So we must consider with IPM. So the first speaker, Dr. Luciana, mentioned about the biopesticide like that. So some microorganisms stimulate the plant, inoculated plant, and the plant shows a systemically resistant against the bacterial and viral diseases like that. So there are several reports about that. So in the world, so there are several reports about the biopesticides. So for example, again in bananas, the Panama disease is very famous and caused by the fusarium. So we can use the non-pathogenic fusarium to control the Panama disease like that. And the vaccination or inoculation with a virulent or attenuated isolate treatment has been not only against virus diseases, but also against bacterial and fungal diseases. So now we must image the combination of vaccines for plant virus diseases. Yeah, because not only fish, but also in humans and animals. So there are many combination vaccines are used now. So the maximum is 6 vaccines are mixed for cows or pigs, like that. And also that we must consider the integrated pest management. However, now we must consider about the integrated plant management, phytobiomes, because the diseases, how to say, the suitable disease, suitable condition, suitable condition for disease growth are not only caused by the pathogens, but also the circumstance, atmosphere, or nutrition, and so on, so on. So we must consider the agriculture with the integrated philosophy. So this philosophy, integrated plant management, guide us to the sustainable development goals in agriculture, I hope. So in that case, so the first in the local or in domestic collaboration is among local governments and universities and commercial companies. So for example, in development of vaccines against plant viruses, the universities, academia studies, academia analyze viruses with molecular biology, and also the local government research institute are how to apply in the field. And commercial company also produce the commercialized products like ampoules or like inoculation method and so on so on. So these collaboration is very important. And also for farmers, dissemination and the awareness of diseases about diseases are very important. And so finally, the second importance is international collaboration. So I would like to say that the Vietnam National Agriculture University and Pachajara University and Utsunomiya University. So we can collaborate internationally and let's fight against our common enemy, plant viruses. So thank you very much. That's all. Yeah, thank you. Thank you for your attention. Hi.
Thank you. Such a fascinating talk and such a timely talk, also connecting with the ongoing pandemic. Okay, so we do have a few questions in the chat here. First, there's a question from Dr. Nono, and I'll read it. So it's two questions. The first one is it possible to grow crops without virus infection?
Sorry. So that is in chat.
Yeah, it's in the
okay.
chat.
Does the next generation have? No. Yes. So for example, if potato or sweet potato or cassava, if we can establish the good attenuated isolate, so we can use that second generation or third generation because viruses can transmit through seed potato. All right. However, usually the seeds, the plant virus cannot transmit by seeds, through seeds.
Okay, yeah, so this is an answer to the question from Ibnu at UNPAD about that. So it can transfer from generation to the next?
Yes. And so for example, in tomatoes, it's very easy to cut and multiply the seedlings. So if you can, you have inoculated attenuated isolate to tomato plants and cut many parts and how to say, multiply on the soil. So we can multiply, yes.
I see, great, thank you. Yeah. Shall we continue through these questions here? I was trying to start from the top in the order that they came. Do you see this one from Dr. Nono
Yes.
Carsono?
Sorry. I have two questions. It is possible to grow crop without virus infection. What?
Yeah.
Yeah, of course, yes. How is the effective ways to protect plants free from viruses? Yeah, so for example, if you have the good protected glass house, for example, so you can grow the plants free from virus infection, I think. However, it's very difficult, especially in tropical countries, because in tropical countries, always, for example, there are insects which can transmit viruses. However, in USA or in Japan, so we have winters and many insects died outside of glass house. So if when we can control the insect in glass house, so we don't have any virus transmission in next season like that. All right. And the next, I understand that the plant immune reactions of plants are not the same as from yes, different. So the plant do not produce any antibodies against the pathogens. Yeah, so that it's very difficult to explain what is the immunity of plants. So that is a big problem in plant science now. Yeah. So the many molecular biologists in plants are researching about the mechanism of immune systems in Japan, in plants, sorry.
Okay. And then there's a question from Anas Zubair. I can read it or, okay. Natsuaki Sensei, I have a question. Is if the mutation occurred, it means we have to formulate against the new attenuated isolated virus?
Yes, yes, yes.
Okay, so that was a clear answer on that. Great. And then I think you answered this question already from Ibnu at UMPAD, that it
yes,
can
correct.
go to the next generation. And then, okay, and then there's another question from Anas Zubair at the very bottom. Natsuaki Sensei explained that the seedling price is $1 higher than an ordinary one. Is it okay for Japanese farmers?
Yes. Yeah. Benjamin, as you know, how much one tomato or one cucumber in Japanese supermarket? So maybe in this autumn, the one cucumber is more than $100. I think so. It's okay, no problem for Japanese farmers. Yeah, of course, that's if you would like to use this technique in developing countries, the price should be less, more cheap, of course. But I think the laborer's salary is also the less.
See. Great. Well, thank you so much for a really fascinating talk, and such great questions from the audience as well. Okay, so I'm not sure. Is there someone, Dr. Yamane, or would someone else like to share some closing words?
Okay. Thank you very much, Dr. Luciana and Professor Natsuaki. Both are related to plant protection and viruses, fungus. Very nice topic today. Thank you very much. And we also thank to all participants today. I'm very impressive. The number of the participants over 500.
Yes.
So our Zoom license maximum 500. So Dr. Nono used the UMPAD license and maximum 1,000. So you are right. Thank you very much, Dr. Nono, and a member of UMPAD. This is our first session. So, but I think that we can do the second seminar next month, right?
Yes.
So let's discuss later the detailed day and time again. So let's, but anyway, thank you very much today. I think it's very successful for your collaboration. Thank you very much.
Thank you.
Oh,
Thank you,
Anas-san.
Sensei. You're welcome.
みんな元気ですね。
Okay, well, thank you so much to everyone for joining today. Very exciting to see such international connections at this time. And thank you.
Thank you, Sensei.
Thank you very much.
Thank
Thank you very much.
Domo arigatou gozaimasu. Thank you
very
for
much.
thank you very much for everybody.
Thank you very
Terima
much.
kasih.
See you then, Sensei.
Yeah.
See you next time.
Hopefully, yeah.
Terima kasih.
It's nice.
See you to the next.
Akhirnya.
Thank you.
Okay.
Thank you.
Thank you very much. You again.
Nono, makasih ya.
Terima
Sama-sama,
kasih.
Bu.
Domo arigatou, sensei.
Terima kasih semuanya ya. Dari
あ、
Usu,
ノノさん
dari
どうもありがとうご
Unwir,
ざいました。
Domo
Yes,
arigatou
hai.
gozaimashita.
Terima
pada
kasih.
Sensei, thank you.
Kendari Pak Anas, dari
Dari
Kendari.
Kendari, arigato, Bu. Thank you, Bu.
Terima kasih.
Halo
Terima kasih.
Oleo.
Halo,
Thank you,
Oh,
Yamane
ya.
Sensei.
ada dari
Oh,
Usu tadi ya, Unwir.
From east to west ya. The member of today's webinar sensei in Indonesia.
あ、いや。
Ya, Yamane sensei, genki desu ne.
Bilang tuh.
Yeah, thank you. Thank you.
Omedetou
Thank you very much.
gozaimasu.
Terima
Very
kasih Bu
acceptable.
Luciana.
Very happy.
Terima
Iya,
Sama-sama,
kasih.
Bu.
ini suara siapa ya? Saya nggak
Sangat
kenal.
senang
Oh,
kami dari Halo Oleo.
Oh,
Kita Halo Oleo
iya,
Kendari.
Oleo from
berapa tahun yang
Manado.
lalu ya kami ke
It's a
sana.
very
South
far.
East Sulawesi.
Southeast,
Sulawesi.
Sulawesi.
Ya.
Sulawesi Island.
Thank you.
Terima kasih,
Dr.
Bu.
Nguyen, are you still there? I was just curious with the background of your picture. It is the university because several years ago we went there to the university, the Vietnam National University of Agriculture, but I think the building looks different. But Dr. Nguyen is not here anymore, I think. Oh, here, you're still here. I'm sorry,
It's
I
different.
cannot recognize you. Perhaps when we went there, maybe I saw you as the organizing committee.
I think Pak Nono, after pandemic COVID-19, we invite our friend from Vietnam also from Usumia visit our university again. Yeah.
Thank you very much. Yeah. Yeah, I really want to visit you.
The next, Yamane sensei.
Yes. Yeah. Okay, thank you very much.
Thank you very much.
I have to leave now.
Take a rest,
Thank you.
Sensei. Yeah.
Good night.
See you then.
Okay, thank
See
you.
you.
everyone.
Thank you.
はい、バイバイ。
Bye bye.
Bye.
Thank you very much. Bye bye.
Bye.
Lili, saya nanya barangkali di YouTube ada pertanyaan nggak kebaca ya?
Nggak ada, Bu.
Oh, nggak ada ya?
Kosong, Bu.
Nggak ada yang ikut YouTube ya, Li?
Nggak ada, Bu.
Oh,
Tadi dulu
oke.
udah cek. Kayaknya masuk semua ke sini karena kan 1000 ya, Bu. 1000 peserta.
Iya sih, Mas. Oh iya, jadi nggak ada.
Jadi nggak ada yang di YouTube.
Nih, kalau udah ada rekaman YouTube-nya bagi dong ke saya.
Oke, Bu. Iya, Bu.
Live streaming Bu Lucy, itu live streaming. Jadi dicek di streamingnya Unpad.
Oh, ada ya? Oke.
Webnya
Mau
Unpad.
buat ini, buat koleksi arsip.
Iya. Kang Sandi mungkin di stop live streamingnya Kang Sandi.
Oh iya, Kang Sandi ya, nanti tolong ya. Saya mah gaptek, nggak tahu, nggak bisa mengakses, nanti minta
Cek ini
tolong.
Bu, di streaming
Kang Sandi.
Unpad ACID nanti sudah ada.
Oke.
Streaming Unpad ACID. Iya.
Oke, semuanya terima kasih nih yang
Nuhun Bu
sudah
Lucy. Iya.
bekerja keras. Saya keluar ruangan ya. Salam buat semuanya. Ini ada mahasiswa saya juga banyak nih, mahasiswa kelas saya yang ikut hadir.